Hi, my name is Sylvere Bastien. I am a bioinformatician at CIRI located in Lyon, in the staphylococcal pathogenesis team led by Professor François Van den Esch. I'm going to talk about a project we conducted that focused on the possibility of strains causing Staphylococcus aureus bacteremia to cause also infective endocarditis. First, few words about Staphylococcus aureus. It is a commensal bacterium in humans, present in about 30% of the population. It can cause a wide variety of clinical diseases, including skin and soft tissue infections, osteoarticular infections, toxic shock syndrome, and bloodstream infections. We were specifically interested in this last type of infection. Most studies have shown that in 10 to 25% of safe cases, a second deadly infection called IE occurs. Given this percentage, we wondered if it could be related to the expression of specific virulence factors. To answer this question, we used two approaches. The first approach was based on genomic analysis, aiming to determine if the strains causing IE were genotypically different from non-IE substrains. The second approach focused on a more phenotypic aspect using animal models. The goal was to observe if the strain causing IE behaved differently in experimental model of IE. Regarding the genomic analysis, we employed three different approaches. We first investigated if there was a common heritable trait. To do so, we measured a phylogenetic signal using the results of a single nucleotide polymorphism analysis. Then, we examined the coding sequence and non-coding small RNAs content. The aim of this analysis was to find a combination of markers associated with strains causing IE, and this was achieved through lasso regressions. Finally, we conducted a genome-wide association study, GWAS, based on k-mers to associate genomic patterns with a particular phenotype. A few words about the strain set. We obtained sequencing data from 924 S. aureus strains, including 274 classified as causing IE. These strains were collected from 4 international cohorts. Concerning the common heritable trait, the variant calling results allowed us to identify nearly 15,000 SNPs. A phylogenetic value was then calculated using Pagel's lambda and compared to 500 values obtained by permuting the phenotype. Regarding the combination of genomic markers, we annotated nearly 5,000 coding sequences and non-coding RNAs. We measured the phenotype genomic correlation using the Cohen's-Kappa regression coefficient. Similar to the common heritable trait, this value was compared to 500 values obtained by permuting the phenotypes. For the animal models, we used mice and rabbits. Our goal was to investigate both the early stages of infection and local and systemic development. To achieve this, we employed two types of models, inflammation-based models and mechanical damage-based models. For the mouse models, S. aureus infection was induced either by an intravenous injection of a strain pool or through catheter placement for specific strain infection. The catheter was placed according to the model, with HS indicating high severity and LS for low severity. We measured adhesion and propensity to cause IE. For rabbit, S. aureus infections were all conducted via the marginal ear vein. We measured vegetation size, counted bacteria from these vegetations, and enumerated bacteria in the blood. Regardless of the results from both genomic analysis and animal experimentation, we were unable to demonstrate any difference between strains causing IE and non-IE substrains. This work suggests that SRE strain variation is probably not the primary determinant of IE. So, whole strain should be considered as capable of causing this common and deadly infection once they enter the bloodstream. I would like to thank all the people who participated in this project. And if you are interested and would like more information and details, you have the QR code to access the poster and our paper. Thank you, and I wish you a great conference.